Himani is a physicist by training, and she'll tell us about her recent work in theoretical population genetics.
Yeah, so as Kavita mentioned, I have a background in statistical physics, but then have been working on various questions in theoretical population, evolutionary. Okay, I think I have to be very loud then. Is this better? Yes, okay. Yeah, so I think there are a lot of, this study of polygenic adaptation tends to attract people with a physics background because, or especially a statistical physics background because, it's sort of, what at its heart, it's about understanding how different genes evolve in a almost not cooperative, but collective manner. So you can't really think of one locus at a time, but we're interested in interactions, in associations between genes and how that shapes evolution in the short run and in the long run. And so here I'll sort of talk about evolution over somewhat long time scales, especially thinking about how genetic differences between populations evolve or how they are maintained, and when this leads to the formation of new species. So I'll sort of start by, giving a very brief overview about, because when people talk of species, they often mean different things. So just sort of setting the terms. And there is a lot of jargon also that people use in terms of barriers to gene flow, and like with a lot of things in evolutionary biology, these are not always sort of, again, people mean different things sometimes when they talk about these. So I'll just try and give an overview and set the terms, give some sense of how I'm going to use these terms. And then I'll try and sort of illustrate the basic ideas in terms of a simple model. And the goal here is to just introduce some heuristics and approximations which give insight into how different loci, why is this a multigenic process, and how do we think about it in terms of how associations are maintained between different genes are maintained or how they evolve. And then I'll try and illustrate this with use these to answer some questions, at least in the context of a simple model, focusing on when divergence is maintained at multiple genes. So when different populations are genetically differentiated at different locations along the genome, and whether this can persist in the face of mixing between populations. I'll then sort of go a bit away from the simple model and start talking about, or just briefly talk about, more complex genetic architectures of differentiation when the differences between species involve loci with arbitrary dominance or with physical linkage along the genome. And sort of finally, I will try to take a step back and say something about effective parameters like effective population sizes, effective migration rates, because I hope to sort of illustrate that a lot of questions in evolutionary biology, especially when thinking about multiple genes evolving together, can be sort of thought about fruitfully in terms of these effective parameters. Though, of course, there are limitations. As far as I know, this diagram is the only figure in the origin of species. That's true, right? Yes. So this was the only figure in the origin of species where Darwin drew this sort of cartoon of how different populations might diverge in this sort of tree-like structure. One could sort of try and focus on any one of these extreme splitting events, the most recent splitting events, and one might end up thinking about species which look quite similar to each other. So this is the famous example of Darwin's finches, which I think also inspired some of Darwin's observations. And then, of course, these kind of species or subspecies are now studied using sort of genomic data, giving rise to some kind of phylogeny. So, these are many, I don't know how many these are, but this is a sort of phylogeny of different finches on the Galapagos. Actually, since this paper was published, I think the phylogeny has been revised, and this is not just because of, this in part at least reflects the fact that defining species is sort of inherently a murky business. So there are different species concepts, different ways people classify individuals into species. There are so-called morphological species, where concepts where you might do a PCA of some sort of phenotypic measurements, and you see these clusters, and those are the species. So many of them are based on sort of some kind of clustering, really. But one of the sort of definition that has proved to be really sort of influential in evolutionary genetics is the so-called biological species concept. This is not sort of useful in practice to define species, but it gives a way of thinking about the processes that lead to speciation. So under this concept, a species is basically a group of individuals that can interbreed amongst themselves, but that is, they can produce viable offspring. But if you consider two such groups, they do not produce viable offspring, either because they just fail to mate with one another, or even if they mate, the offspring, the hybrids they produce may be sort of inviable, infertile, or just fail to survive, or yeah. So, the term people often use is that these groups are groups that are reproductively isolated from one another can be considered species. But of course, this is a loose definition. We would ideally like a quantitative measure of this kind of reproductive isolation. And so one way of doing this is to take a genetic perspective, really. And but before that, when we think of different subspecies, as we were saying, that there are often morphological differences. So in this cartoon taken from this review, there may be differences in sort of coloration, differences in song here, and when these two different species or different subspecies produce an offspring, this individual may be somewhat intermediate, and it may fail to occupy any of the available ecological niches. It may not have good camouflage. It may have song which is not recognized as a mating signal by most of the individuals here. So this, of course, at an ecological, this is, in terms of the processes that we can observe. But if one now thinks of the genetic basis of these traits that are sort of being selected against. So in this example, so because this is a toy example, it could be that the genetic basis is simple. You have basically these two loci here on this chromosome, one here and the other here, which underlie these genetic differences. So in this first population, you have the blue variant, which leads to this sort of phenotype. In the second population, you have these red variants, these red alleles that lead to this kind of phenotype. And if there is a hybrid once in a while, it will carry both red and blue variants here. And if this hybrid is unfit for whatever reason, it will have fewer offspring than, say, either the blue ones here or the red ones here. So these, it will pass on. It's less likely to pass on its genes, its alleles to successive generations. And this is not just true of these genes, these alleles that are responsible for the differences between, which are phenotypically, which underlie these selected phenotypes, but this is also true of neutral alleles which have no effects on fitness. So in some sense, one way of thinking about this is that the sort of exchange of these neutral alleles is also inhibited. But of course, we also know that in sexual populations, different bits of the genome evolve independently. or more or less independently over long time scales, because these are sort of separated by meiosis. So this effect where gene exchange is suppressed is stronger in regions that are closer to these loci, because they're more likely to, so for example here, alleles on a different chromosome are sort of more likely to, if this hybrid ends up somehow at least mating with the, maybe it ends up producing at least one or two offspring, then these alleles may end up on a background which is sort of favored in this first gene. And this means that it gets decoupled basically from these divergently selected loci more easily. So this sort of summarizing that here, that gene exchange is reduced at neutral loci that are linked to loci that are selected differently in the different populations. And because these selected loci sort of inhibit gene exchange at nearby locations, they're often also referred to as barriers to gene flow. And one might quantify this by defining an effective migration rate. So one might ask that, if I have a neutral allele here in the middle of these two selected loci, what's the rate at which it gets transferred to basically this resident phenotype or the resident genotype here, which has the blue alleles. It comes, it sort of enters this deme on with these red alleles, which are harmful, but by a few, in a few generations, it might recombine away and find itself on the resident genotypes, in which case it doesn't have a selective disadvantage. So, so one way of sort of thinking about this sort of reduced gene flow is to ask what's the rate at which these immigrant alleles, neutral alleles, are transferred to the resident genetic background. So 2 points. One is that this effective migration rate is going to be different for different regions along the genome. If you are sort of far away from these barrier loci, these selected loci, then you can recombine away much more easily, and the effective migration rate is close to the actual migration rate. If it's you're close to these barrier loci, then of course there is this effective migration rate, this rate of exchange is reduced a lot more. And the second thing is that if you want to quantify how much it's reduced, then it depends on how unfit these hybrids are, and not just these hybrids, but also other recombinant offspring, which might now carry just not two bad alleles, but let's say one bad allele. So you need to know the relative fitness of all these recombinant hybrids or recombinant hybrids and recombinant sort of descendants to quantify so these fitnesses are going to determine the effective migration rate. In this toy example, I've talked about two barrier loci, but it's quite likely that there are many such barrier loci, many such loci that underlie trait differences for ecologically, for traits that actually may be selected differently across species. So, there are several lines of evidence for this. One is just a general expectation that most of the traits we know are quite polygenic. Of course, there's evidence from jivas, but also, the evidence from sustained response during artificial selection. The other sort of signature is, but you can also see signatures of polygenic barriers, let's say, in genomic data. So in this example, this is data from Heliconius, which is this very charismatic, closely related species of butterflies in South Central America. And what is being plotted here is the proportion. So they have these windows along the genome, and in each window they measure how much of, what is the admixture proportion. So you have these two different species, and between a species pair, you can measure the admixture proportion using various, so you can quantify it using various statistics, and then ask how does this admixture proportion, what does this admixture proportion depend on? So here it's being plotted against the local recombination rate in that window, and there is this sort of modest positive correlation. So the higher the recombination rate, the more mixing there is. Similarly, for another species pair, this is a different species pair within the same complex, and one sees the same sort of observation. And sort of one hypothesis for this is that, I mean, one way of explaining this, although there are complexities, I think, is that if you have many, many barrier loci, then most neutral sites are going to find themselves in the vicinity of one such selected locus, and they'll be affected in terms of this reduction in gene flow. But in windows where the recombination rate is high, this reduction is lower, and there's more exchange, more admixture. So this explains this sort of could explain this sort of positive correlation between admixture proportions and local recombination rates. This sort of signature is often used also in not just looking at divergence between species, but also diversity within species, where the effects of linked selection are sort of become weaker, the effects of hitchhiking or any kind of linked selection in local windows, which are windows with locally high recombination rates. So this is all to say that there's a way of sort of thinking about this process of divergence quantitatively in terms of how much gene exchange there is along the genome, that this sort of depends, this is going to vary along the genome and depend on the fitness of various intermediate hybrids. And also importantly, now that we start quantifying it, it's not going to be 0 or 1. So reproductive isolation is in some sense itself on a continuum. So, what is a good species is a bit murky for, especially for closely related species. Think through some of the consequences of having polygenic barriers. So we've also seen that with a lot of polygenic traits, individual loci can have very weak effects and are weakly selected. So if any of these, any such locus can be easily swamped, that is to say, if you have two populations, and different alleles are favored in the different populations. So if the selective effect, the strength of selection favoring one allele is S, typically we expect that at migration selection balance, if we just consider any one locus, you can maintain differentiation between these two loci only as long as selection is stronger than migration. So this could be quite a stringent condition if selection for any individual locus, because with polygenic traits, individual loci often have. But now, if we think about the aggregate effects of selection at many genes, what determines the rate of exchange at any one such locus along the genome is not the actual migration rate, but the local rate of exchange at that location. So and if this is much lower than migration, then, in the actual raw migration rate, then potentially you could maintain adaptation across a broader range of conditions. Yeah? I'll sort of make these arguments a bit more precise later. And the important thing also is that there's a positive feedback here. So if there is weak differentiation at many, many loci along the genome, this means that there is strong genome-wide. This could still be consistent with strong genome-wide differentiation, in the sense that if you think of a polygenic trait that's determined by many loci, you could have significant phenotypic differences maintained by relatively small allele frequency differences between populations, in which case, the relative fitness of hybrids can be quite low, because hybrids are sort of intermediate between these two highly diverged phenotypes. And if the relative fitness of hybrids is low, this further reduces effective migration rates. because most of these genes coming in on migrant backgrounds are not passed on, most of these alleles, and if effective migration rates are low, this further increases differentiation or can increase differentiation. So there's a positive feedback which we sort of need to think about when we're thinking of how these different barriers are maintained, how polygenic barriers are maintained. So I'll try to now make this a bit more precise using a simple model. So let's imagine that we have two ecological niches represented in these blue and reddish tinges. And I'll just describe in a minute how these ecological niches are distributed across space. And let's imagine that alternative alleles are favored in these two ecological niches at several loci, so let's say L loci. And I'll also, for the time being, assume that these are unlinked, that is, by which I mean that they may be on different chromosomes, or basically, if you think of recombination rate between adjacent loci, between any two loci, sorry, not adjacent, then R is half. Okay. For the time being, because it's a simple model, I'm also assuming equal effect loci. So if you are, yes, so the locus, selective effect per locus is S. And now fitness is sort of depends of any genotype, depends on whether it carries, if an individual carries mostly blue alleles and it lives in a blue sort of region, then it's quite fit. And the loss of fitness really is determined by the number of locally disfavored alleles that it carries on the genome. So it's essentially a model of directional selection, which is divergent across these different habitats. Yeah? And the sort of geometries of how these niches are distributed, of course, in reality would be arbitrarily complex, but to get some insight, one can look at simple models. So I'll sort of talk of these two models, one where either you have this mainland, which is sort of perfectly adapted, and then we ask what does, where basically the blue alleles are all favored, and then we have a small island, a small peripheral population, and ask how, where the red alleles are favored, and there could be migration, sort of asymmetric migration, so from the mainland to the island, but not the other way around. And there's also population, so I'm going to assume that the island has a certain fixed population size n and a certain rate of migration from the mainland to the island. The other sort of caricature or other toy model we can think of is the so-called infinite island model, which was introduced by Wright, where you imagine that there are these different demes which are all connected to each other. So any individual can migrate to any other, any individual from any deme can migrate to any other deme with equal probability. So basically with rate m, an individual leaves its home deme and then finds itself across any of these other islands. And I'll assume that some fraction of islands have this blue habitat or blue niche, and the others have this red habitat or red niche. And this need not be half and half. It could be some arbitrary fraction. And sort of one of the questions I'm interested in is asking, what conditions facilitate polygenic divergence or polygenic adaptation to a rare or somewhat rare ecological niche. So just to, it's the fraction of islands with one of the environments. So that 1 minus rho is, they don't change over time. So if you imagine that there are 100 islands, 20 of them have one kind of environment, 80 of them have one kind of environment, so rho is 0.2 because in 20 of them a different set of alleles is favored, and 80 of them a different set of alleles is favored. Okay, so there are a few parameters in this simple model, and one of them is the number of divergently selected loci, the other is selection per locus, then the migration rate, the population size. I should say that each of these are equally sized populations, and migration is sort of, these arrows are at the same rate of migration. So also in this model. So there's number of loci and selected selection per locus, this sort of more parameters, and these three parameters that describe in some sense the landscape or weather and demography. So you have migration rate, population size, and the fraction of alleles in the two habitats, fraction of islands in the two habitats or niches. So I'll use this to illustrate, the basic heuristics that one can use to understand this kind of model, and then go on to more complex, just briefly mention more complex architectures where these loci are linked along a chromosome and where selective effects vary and where dominance effects, there could also be potentially dominance. So within this sort of simple model, I want to ask a few questions. So first is, beyond what level of migration is adaptive divergence lost? And for the mainland island model, as I said, there's a simple criterion. If populations are very large, then we expect that basically local allele frequencies of the adaptive allele are, so I can write these as, this is for the locally bad allele, so locally deleterious allele. But of course, so once M exceeds S, you are basically fixed for the locally deleterious allele. But what happens when there are many such loci? One could argue that it must depend on the effective migration rate, but we don't actually know what the effective migration rate depends on, So I want to ask, how does this change when there are one versus many selected loci? How does genetic drift affect these thresholds? What happens when if you have a sort of infinite island model, a metapopulation, what happens in one of the niches is very rare? And also, often we don't really know what loci are selected, so we want to have some measure of these effects on neutral diversity along the genome. So these are the questions I'd sort of like to partially answer. I'll just use the next two slides to illustrate the basic idea of the theoretical approximations. So the simplification is that when we think of highly polygenic barriers, then roughly speaking, if you have two divergent populations, they differ at 100 selected loci, hybrids are going to be somewhere midway, so they'll differ at approximately 50 loci from one or either of the two populations. So they're going to inherit half of the migrant parents' genotype and half of the resident parents' genotype. And of course, there is some sort of segregation variance within, well, not for F1 so much, but for later generations. I'll just come to that. And now, if migration is low, and by low, I just mean that it's not that NM is low, that's not the number of migrants is low, but M is low. So migration rate per generations are somewhat low. Then very often, most often this individual, this F1 individual will mate with another individual who's resident to that population. So this hybrid will end up mating with the resident individual, another individual who's sort of well adapted. In that case, now the next generation of descendants will inherit about half of these blue alleles, so a quarter of blue alleles or the wrong alleles on average, and 3/4 of the resident genotype. So there's a half, in every generation of successive mating, the fraction of deleterious alleles that you carry halves approximately. And you can sort of go on and thinking about this. So it's just a geometric series. Migrants are fully deleterious, or at least in terms of their relative fitness. Hybrids half as deleterious, backcross is 1/4, and so on. So if we now think of the effective, if we think of any focal allele on the genome, and this allele is far from these selected loci, we can ask what is the fate of this allele? How is it transferred? But what's the probability that it will eventually, it comes in on this migrant genome, but it will eventually end up in one of these red genomes, one of the resident genomes. So we want to basically ask what's the probability that it flows from this background to this background to this background. Yeah? And this will basically just be determined by the probability that the migrant and this F1 and this backcross one, that's this individual here, and has offspring. So there is this sort of backcross formula, one could call it, where we say that for an unlinked allele, at least an allele that's sufficiently far, not in the immediate vicinity of any locus, this reduction in effective migration rates just depends on the relative fitness of all these hybrid and recombinant offspring. So this sort of gives us some handle, but of course we still don't know what the relative fitnesses of these individuals are. But we have a sense that this should depend on how different these two, how different these two genotypes are. So if there's a lot of swamping, if there's a lot of gene flow and populations are becoming similar, then these relative fitnesses will be high. Because they're not very differentiated, but if these populations are very diverged, then these relative fitnesses are low. So the basic idea of this sort of analysis then is to calculate these things self-consistently. So I ask if I knew what the relative fitnesses of all these individuals was, I could just calculate the genome-wide ME at not in the vicinity of a sort of selected locus, but at anywhere sufficiently far. And using this sort of formula, if I knew ME, what I'm going to do, and this will sound a bit ad hoc, is we know how any single selected locus behaves. We know how any single neutral locus behaves in terms of, there's well-developed theory for the last 100 years, or I don't know, at least somewhat in that order of time, where we know that if you have, we can sort of write down various things, at least the equilibrium allele frequencies and equilibrium allele frequency distributions given selection, migration, mutation, drift. And so the basic idea is that I'm just going to replace M with ME in this sort of theory. If you're interested in the maths, it looks a bit like this, not infinitely complicated. But the key point here is that if you see, for example, here, I'm writing P as ME by S. So the allele frequency, the locally deleterious allele, has a frequency that is determined now by the balance between effective migration rate and selection, not migration and selection. And if I now know allele frequencies at various loci, I can sum them up to get how many alleles are different between migrants and residents. I can then use that sort of in this kind of formula by noting that if migrants and residents have a certain number of alleles that are different, then the F1s are going to have approximately half as many that are different, and so on. And when one does this, one has essentially three equations which all depend on each other. So here the Y's, the number of alleles, these depend on these allele frequencies. Then the effective migration rates depend on the total number of alleles that are different, and they determine in turn these allele frequencies. So it's a circle, and one can just solve it in a self-consistent manner. And so this thing that I sort of said here, that replace M with ME, I mean, it sounds a bit ad hoc, and of course one can ask, when does it work? When is it justified? So this sort of replacement is really justified when these kind of associations that we are talking about, that there's a neutral allele, it's on a migrant background, it ends up on an F1, and so on. And then over, let's say, 10 generations, it's ended up on a resident background because of successive recombination. And of course, this will happen only when it's loosely linked or unlinked, ideally, to any specific selected locus. So once that happens, over 10 generations, this sort of process of decoupling has happened. And if the other processes that I'm talking about, migration, selection, they are not occurring, they're not producing such drastic changes over 10 generations, then there's a natural separation of time scales. So these associations are broken down much faster than allele frequencies change due to any of these single locus processes. In which case, it's as if there is just a rescaling of the migration rate. Yeah? Genealogies at any such locus, selected or neutral, are not really affected over long time scales. Genealogies which depend on longer coalescence time scales are not affected usually. So I'll come back to this point when talking about effective parameters in general. So this is basically the theory that I'm using here. I've illustrated these formulae that I've written here are actually valid for the mainland island model, but one can do something similar, only slightly more complicated for this infinite island model, but I won't go into that. Yes, I mean, but it's a geometric series, so if you sort of think of this, no, yeah, because if you think of, let's say the migrant and the resident differ at Y alleles, so the fitness of the migrant is then this. The fitness of the F1 hybrid is approximately this. Of course, one can vary about variances and so on, and they'll contribute order S squared terms, but just to a first approximation. Then the next generation is, yeah. So then you basically have E minus S by 1 plus half plus 1 by 4 plus, so which is E to the minus 2 S1. Yeah, so that's why you have this factor of 2 here in this first year. It's entering here. You have to sum over all the.
L is large enough.
No, it would not work so well for 2, 3. I mean, with 10 it works, but 2, 3 is a bit, I think, pushing it. Okay, so, no, it does work for 10. I don't have 2, 3 here, so I'd forgotten. 1 is just, diffusion approximation, so there's no approximation, but I don't have anything between 1 and 10. Okay, so I'll just go through quickly a few of the results of this sort of model. So what I'm plotting here now for the mainland island model is the expected frequency of the locally favored allele as a function of migration rate, where I'm scaling migration rate by selection because, and this is for a population where drift is important, so NS is 2. And so the different colors represent situations where you have 1 versus 10 versus 20 selected loci. The symbols here are simulations, and the lines here, these lines are the theoretical predictions. So the first thing, of course, to see is that the theory matches simulations very well. So it's sort of capturing the essence of these complicated multi-locus interactions. The second thing is that at any given migration rate, if you have many more loci, the expected frequency of the locally favored allele is higher because you have all these other loci that come in. Any one allele comes in along with all these other divergently selected loci. So the selection that it experiences is not just due to its own effect, it's because it comes in as part of this highly unfit genome, and there is this sort of collective elimination of these deleterious alleles because the hybrid just does badly. No, it's maximum recombination, R equals half, so unlinked loci. Without recombination, of course, it would be as if you have a single locus. All of it would be, but with effect size S times L. Okay. The other thing we sort of notice is that these thresholds become very sharp, because as migration increases, there's hardly any change in divergence, and then there's this sudden collapse and everything just, this mainland and island suddenly become very similar. So this sort of sudden collapse is quite characteristic of polygenic divergence because, if we sort of go back to this kind of picture that we have, there's a positive feedback, yeah, that even if there are slight changes at divergence levels at individually at many loci, together, if there are many such loci, together they can involve quite a strong effect on overall divergence, which can have a strong effect on fitness of hybrids. So slight changes are amplified, per locus can be amplified by just having many, many more of these loci. And these kind of positive feedbacks give rise to these sharp thresholds. I want to emphasize that we're also accounting for drift here, and I'll sort of show that a bit more in detail. So now I'm sort of fixing the number of loci at 40. So you have 40 loci along the genome, and the different colors correspond to different population sizes of this island. And again, the symbols are simulations. These solid lines are full theory. And for reference, I also want to show the dashed lines, which tell you here, for example, what the allele frequency would be if it was just a single locus with that level of NS and whatever, no. So again, one sees that as you have larger populations, of course, selection is more efficient at the level of individual loci, but also these collective effects become stronger. That is, you have these sharper thresholds. This is because, again, we saw that there's a positive feedback between divergence at individual loci and how it increased divergence at individual loci has results in increased divergence at the level of the trait, reduced effective migration rate, and so on. So we sort of see this kind of sharper thresholds in larger populations and for loss of migration. Well, actually, this is for stochastic. So you have to, that's only for the deterministic case. For the stochastic model, you need to use Wright's distribution. But with ME, where this ME depends on other loci. I don't have that here, but one can easily work that out. I mean, I do I have it? I have how...
I guess it should be the way you're showing for deterministic.
Yeah, so there's just one curve here for the deterministic, but this is the frequency of the locally favored allele. So yeah, I mean, this would be, yes, because S is fixed. So yeah, you can, for the deterministic case, you can also think of this as how ME changes with M. Yes. Okay. And this is just, the details here are not so important, but I also wanted to say that, so far I was looking at the expected frequency of the locally favored allele, but actually one can predict the whole allele frequency distribution at any one locus. And this also matches the theory very well. And this is sort of a bit non-trivial, I think, because, when we often these lower dimensional summaries of the whole allele frequency spectrum. So if we think of something like heterozygosity, it just depends on, you trace two lineages back and you ask when do they coalesce, or when do they find a common ancestor. That's what determines heterozygosity. So that's not entirely, yeah. So I mean, to say that something like heterozygosity or allele frequency is accurately predicted by effective parameters is one thing, but to say that the whole allele frequency distribution is accurately predicted by an effective parameter means that the underlying genealogy is not changed, really. It's just rescaled in this regime. Okay, and I'll just spend a couple of minutes to say something about, so this was all with mainland island. One can do something, make similar predictions for meta population where some fraction of, so in this case, 10% of islands have one niche and 90% of islands have another niche. And the equations are a bit more complicated, but one can still sort of make predictions here. So the different symbols are 10 versus 40. Is that true? Yeah, 10 versus 40 loci. More loci means, so if you look at blue squares versus blue circles, more loci means sharper thresholds. And then you also have strong, the blue versus black are larger population sizes, but still, not so large, so drift is important here. And again, one can see that both with higher populations and more loci, you have shifted and sharper thresholds at which adaptation is lost. So, there's more one can do, which I don't want to get into right now, but one can ask, what are the conditions under which you can actually maintain local adaptation over much larger rates of migration, and one can generate predictions for that, and I can talk about it later if someone is interested. But I do want to say, this is not directly related, it's not a polygenic effect, but I think it's quite interesting, so I'll say a bit about this. So people often look at, we don't really know what loci are selected. If you have a bunch of genomic data, if you have a bunch of sequences, you have no idea what loci are selected, but what one will measure typically is something like diversity or heterozygosity along the genome, or divergence, pairwise differences within a population or between two different populations. So what I'm plotting here is FST between two pairs of, between two arbitrarily sampled subpopulations within this metapopulation. And I'll just write down what this is. So this is where pi within is the heterozygosity within, or the diversity within any island, and pi between is the sort of number of pairwise differences, average pairwise differences across between two individuals sampled from the two different populations. So as we expect, so it's a measure of relative differentiation in some sense. So as one would expect, this as there's more mixing, diversity within populations and divergence between populations looks very similar. So FST here, which is essentially the difference between the two, declines with increasing migration and increasing mixing. But of course, here we have, different, you could sample the two demes quite differently. You could sample pairs of demes, pairs of islands within the same habitat or between different habitats. And actually this sort of relative differentiation, this is highest for FST-RR, which is the FST between two islands, both of which belong to the same habitat. So there's no adaptive divergence between these, but they both belong to a rare habitat. So if I have a population, metapopulation where 10% of islands belong to one habitat and 90% to the other, then I'm sampling two subpopulations, both from the 10% habitat. And in that case, actually, this FST is highest, which sort of looks quite counterintuitive, but have I, no, this is correct, right? It is pi within, yes, okay. So the reason really is that, often when people look for an increased FST as a sign of species, interesting regions which are differentiated, they're assuming that an increase in FST must be because, it's the number of differences between, no, it's the other way around, no? No, sorry. So it's because, I got mixed up. So if you have more differences between two demes, then FST should be higher. Yeah, it should be this way, I think. Sorry. After lunch. So often people assume that, or people used to assume that increased FST reflected higher differentiation between two populations. And then at some point it was realized that, well, it could also indicate lower diversity within populations. Yeah, so reduced, and this people assumed could be because of sweeps or background selection or whatever. But interestingly, and that's also what is driving this high FST here. There is reduced diversity within populations, which one sees here. So pi between is the same for all these different comparisons, but pi within is lower here. But the reason one has lower diversity within populations in this example is not because of background selection or sweeps, but it's actually because of local adaptation, because you have these, most individuals, if you have a rare niche, most migrants coming in to this rare niche come from the other habitat. So effective migration rate is reduced quite strongly, for these islands which are within this rare niche. And the lower this inflow, the lower is the diversity within any of these islands. So you have this rather sort of counterintuitive result that this sort of measure of differentiation is actually highest for islands that actually show no adaptive differences. Yes. No. This is really a consequence of having a rare niche and a common niche. So it's a consequence of this spatial structure. So most of our models, we assume symmetric models. There's one island with one habitat, one island with another habitat, equal frequencies. So it's a consequence of the fact that when one of the niches is very rare, but one of the niches is very rare, but still large enough to sort of have, sustain enough diversity across the niche as a whole. So, different subpopulations within the niche may be fixed for different alleles, because there's still, it does encompass many different local populations. But if it's much rarer than the other niche, so if it encompasses 10 local populations versus 90 of the other, then there's a sort of asymmetry in effective, the rate of inflow, effective rate of immigration into this rare niche, because most of individuals coming in are from the common niche. They are sort of poorly adapted. So there's very little coming into any one of the teams, which sort of reduces the heterozygosity within any team. Because this, yeah.
So you're explaining why do we see that's what I mentioned in the...
Yeah, I mean, so if you look at pairwise differences between islands, yeah? So let's take two of these blue islands and look at how, so pairwise differences between islands are not very different, it's the diversity within that's being reduced. Okay, so this was all still for neutral, sort of simple models with unlinked loci, equal effects, all of that. Now, but of course, we know that that's not what genomes look like. I won't actually go through these formulae, but I would just want to say that one can write down expressions for effective migration rates also under more complex architectures. For example, if one has arbitrary dominance and selection, these are of course approximate formulae, but actually they do quite well, as I'll show. One can also sort of think about these barrier loci that are now linked along the genome and work out the effective migration rate at any one such locus. This will again vary along the genome. One can also generalize this sort of general self-consistent argument to other forms of, other forms of mating, especially assortative mating and sexual selection, for which Parvati has a poster. And the same sort of theory applies. I'll focus on the case of linkage because that's most interesting. That's what we see along the genome. So let's just, I'll come to a sort of more concrete example just to sort of see where the theory breaks down. It basically, I'm averaging differentiation along the genome and just plotting as a function of m by s. But the different colors now correspond to different levels of recombination. So red is where the typical, if you think of a genome, and there is some typical rate of recombination R between adjacent loci, then red, and if S is the typical selective effect per locus, then it basically, the theory applies for loose linkage. So basically anything where R by S is greater than 5, but it sort of starts breaking down once, well, it definitely breaks down for R bias equals 1. So when recombination is as strong as linkage. Because if you remember this sort of argument that I made, that let's just replace ME with M, required that these associations between loci break down much faster than evolution acts at a single locus, and that will not be true once linkage is strong. So I'll just go through this one example because, well, it's a bit like what genome scans look like. I mean, okay, this is with a lot of averaging, so it's much less noisy than a typical genome scan. But so what I'm plotting here is an example where I have 20 loci along a chromosome of map length 1, so 100 centimorgans, let's say. And so R bias, and these loci have different effect sizes, so they're unequal effects, and R bias bar is 5, so this limit at which it starts breaking down, so somewhat loose linkage. In so I'm plotting the adaptive divergence, that is the allele frequency differences between the mainland and the island at each of these loci, which are different because the selective effects are different. And I think one of these, either the squares are the theory and these lines are the simulations, or the other way around, sorry, I've forgotten. And I'm also plotting neutral FST, so FST at neutral loci along the genome, and again, I think one of these is theory, and the solid versus dashed is theory versus simulations. And this line here is the FST that you would have if there were no barrier loci. This is the neutral FST that one would have. And these two different figures differ in whether, first, this is a lower level of migration, this is a higher level of migration. This is 1.5 migrants per generation, this is 3 migrants per generation. So again, we see that, this sort of theory, which is really, it's of course done numerically in the last step, but it's all in terms of the simple effective parameters. It captures this complex variation quite well. And so just to sort of say, this is, when we have these barrier loci, there's the average level of differentiation, neutral differentiation along the genome is increased, that this baseline is increased from here, but you also have these peaks corresponding to strongly divergent loci. I've also marked some of these loci with these stars. So that's just to say that these are the loci that if they had been just with, this, with the selection coefficients that they have, if they just existed independently, these would have been swamped. That is, they would have lost divergence. But because they exist with all these, as a set of different, embedded in this genome, and they're affected by all these other loci, they are able to maintain relatively high levels of divergence. So they are able to, this is the fact that these loci show strong divergence, or at least somewhat strong divergence, is really a consequence of interference between loci. And one can do the same sort of thing for a higher level of migration. Now the overall differentiation is lower, there are fewer of these peaks, but again, you have these loci that, especially this peak, let's say, which would not have existed just by itself, which only exists because of all these other effects, all these other loci. So just to sort of conclude here before I take a slightly broader perspective on effective parameters. So we have a sort of situation where, we're looking at these somewhat simple models, but, still allowing for various complexities in terms of heterogeneous effect sizes, dominance, loose linkage between selected loci. And this sort of approach, the self-consistent approach of looking at effective, which is defining all this, this theory developed in terms of effective migration rates, can capture the effects of polygenic barriers, predict the effects of all these loci along the genome quite accurately. And also these can be quite easily sort of generalized to include genetic drift. So all the examples I showed you were all with NS of the order of 2, 3, 4, so drift being very important. And polygenic barriers. If you have many, many loci, this results in sharper and shifted thresholds for loss of adaptation, and this effect can be especially significant in rare niches. And there are also some mysteries, or some sort of, at least for me, counterintuitive results which are related to neutral FST, which don't really have to do with polygenic divergence, but the fact that if you have these asymmetries in how niches are distributed, then things don't necessarily, there can be somewhat counterintuitive effects, which also I think highlights the importance of going beyond very symmetric models and very simple geometries, just one step beyond to see how much remains robust. So this is all sort of to do with work, in some sense my own work, but I thought it would be nice to, at least that's the instruction we got from Sam, to take a broader perspective about how this relates to some of the things we've been talking about. So, I guess we've all seen this a lot about effective parameters, especially in Mike Lynch's talk, where often, even when we have somewhat complex demographies or linked selection, we can often still summarize it in some way using an effective parameter. But sort of what I would like to argue here is that these effective parameters are especially meaningful when there's a separation of time scales. So in this, if we take the simple example where there is a population which cycles between high and low population sizes, with spending equal amounts of time in both over some fast time scale. So this could be a seasonal fluctuation. Then, provided that population sizes are much larger than this sort of demographic time scale, then of course one can define an effective population size, which is the harmonic mean between these two. But in this case, provided this holds that the coalescent timescales are much larger than the demographic timescales, there is if you were to look at individual genealogies, these would look essentially the same. If you look at the sort of coalescent trees or whatever, then these would look essentially the same as if it was a constant sized population. You don't have this excess of rare alleles, or you still have these predicted side frequency spectra, provided there's a separation of time scales. So this is quite powerful because it's both powerful and depressing. Powerful because it allows us to use the same sort of theory, which is well developed to predict things. Depressing because there's no way we could sort of infer these kind of fast processes from genomic data. So this is kind of clear it's sort of intuitive with these sort of demographic changes. But as we've also seen in this workshop, one has a similar effect if there's selection at linked sites. So, I was right now talking about selection at linked sites because of divergent selection between populations, but essentially one has the same effect if there's, within a population there is fitness variation. And there's this classic result by Robertson in the 1960s that in this case, effective population sizes are reduced by a factor which essentially depends on how much heritable variation there is in fitness. And this factor of 2 kind of comes from the same arguments I was making here, which is that if a neutral allele finds itself on a highly polygenic on a background which is sort of fitter than average, and if this background is fitter than average because of many small effect alleles, in the next generation, this allele will be transferred to another background which will be roughly half as fit, because it only gets half of the parent's sort of genotype and so on. So it's as if, so you sort of have this formula, but effectively what's happening is that these allele frequency fluctuations are larger than, and they're also a bit correlated. So because if you find yourself on a fit background, you'll stay on the fit background for a while before you sort of get transferred by recombination and find yourself in a different background. So, but also in this case, as long as these associations between the neutral locus and any background are transient, genealogies at the neutral locus are again described by standard theory, standard coalescent. It's worth saying that often people use effective population sizes even when fast, this sort of process of linked selection is not fast. They use this as a way of saying, well, look, diversity is lower than what we would expect by a certain amount. So let's encapsulate this using an effective population size. But this can be a bit, I mean, it's then sort of useful to remember that this doesn't still hold. One doesn't still, you know, the genealogies are not described by standard theory, or effects of selection, you know, are not described by standard single locus theory. It's not just a question of rescaling things. So, you know, these effective parameters, thus are, I think, especially useful over long time scales, because in the long run, any of these associations, you find yourself on a fit background, so your, you know, your dynamics are correlated, but you know, this sort of breaks down relatively fast, and then you're on an unfit background, so it might look quite random over long time scales, unlike drift, which is always random and always uncorrelated in time. But so over long time scales, of course, one cannot distinguish between these two processes just based on, say, a single allele frequency, you know, the statistics of allele frequencies. But over short time scales, this may still be possible. And there was a nice paper by Buffalo and Coop a few years ago where they sort of showed that if you looked at allele frequency changes in a sort of across successive generations, and you have a situation where, you know, there's a lot of fitness variation. So if allele frequency change is for a particular allele is positive, and this is because it's associated with a fit background, then it continues to be associated with a fit background for at least a few more generations. So if you look at allele frequency changes between, say, generation 0 and 1, and if it's positive, then between generation 1 and 2 is also likely to be positive, which results in a non-zero covariance between these allele frequency changes. And of course, if you now look at not between 0 and 1, but 10 and 11, this may no longer be correlated, because by then this neutral allele, which happened to be on a good background, has recombined away, let's say. So they sort of showed that especially with weak linkage, you might be able to pick up this kind of signature by looking at covariance of allele frequency changes across, you know, at short times. And one of the things that they applied this to was, you know, data from experimental evolution, where also for Neda and Christian's experiments, where they sort of saw these kind of signatures of linked selection. And this sort of becomes quite important because very often, you know, with polygenic selection, we cannot actually distinguish, we cannot actually infer selected targets. And in fact, they saw these kind of signatures even when they removed all the windows where there were obvious selection targets, and then they reanalyzed the data without these windows and saw that actually there was still this sort of positive covariances, which indicated that, you know, even the rest of this background was evolving with some sort of, had some effect of selection, polygenic selection. So just to sort of conclude, and I hope I'll finish before 1.5. So this is really at the heart of this kind of self-consistent theory, that if linked selection is a fast process, that is, you know, associations break down much faster than allele frequencies change, then its only effect is to rescale population size or migration rate. If I'm thinking about fitness variation within a population, then it basically rescales effective population size. If I'm thinking of fitness differences between two highly diverged populations, you know, then I'm thinking of rescaling of effective migration rate. And when this happens, then, you know, effective parameters are especially meaningful because you still have the same sort of genealogies, the same sort of behavior, but just over a faster time scale, whether at a neutral or a weakly selected locus. And this is a general recipe for constructing self-consistent theories, because you basically use single locus theory plus effective parameters to predict whatever you want at that level, allele frequencies, heterozygosity, whatever. And then you aggregate across loci to predict mean and variance of polygenic phenotypes, for example, fitness here. And then effective parameters, whether NE or ME, are typically related to fitness differences between groups or fitness variation within groups. So this sort of sets the scale for, sets the, you know, stage for a self-consistent theory. And I gave one example of that, which was related to divergent selection. But there's also a very nice example, or I mean, it's a nice example, but I'm not so sure about how this can be used for inference, where, so there's a recent paper which also looks at this, which builds on this paper, where they use the same kind of argument to actually work out the effects of weakly deleterious alleles on diversity. So we know that with strongly deleterious alleles, there's background selection, reduction in diversity at nearby sites, but this is the effects with weakly deleterious alleles are more complex. So how can we get at that? So again, if we knew any, we could predict. The thing with weakly deleterious alleles is one of the issues is that they can fix quite easily in the population. So what these people do is to use single locus theory plus any to predict fixation probabilities of weakly deleterious alleles. Then they they sort of aggregate across loci, across the entire genome to predict fitness variance, and then they use this to predict any, and so on. So again, this is a self-consistent way of doing it, and they find rather these good predictions for, so here you see how much the genome-wide levels of diversity are reduced relative to the neutral expectation. In gray is the classic background selection theory, that it predicts a much stronger reduction, but once you have many weakly selected mutations, so there's a large U by S, then this sort of, because of selective interference, this kind of reduction in diversity, these hitchhiking effects become essentially, or rather these alleles are not efficiently eliminated, and the reduction in diversity is much weaker, and this is very well predicted again by their theory. The total fitness variance is also much lower than what one would expect under standard background selection. So again, this kind of theory predicts all of this pretty well. So, so this kind of self-consistent theory basically allows you to get at regimes where there's a lot of interaction between loci. And I mean, to me, the sort of nice thing is that these descriptions are very economical, because if you sort of think about it, it's all based on allele frequencies, yeah? Because I'm just using ME to predict allele frequencies, aggregating across these to predict trait values and trait variances. And so there's no mention of LD or pairwise LD or 3-locus LD or anything, but what it's capturing is really the effects of LD, the effects of associations between these different selected loci. So it's a sort of very economical description in terms of just allele frequencies, which gets at the fact that there is interaction between loci. And this sort of, in fact, in this paper, which I mentioned, then they use this kind of self-consistent theory to build an sort of inference framework. But I think because these descriptions are sort of quite economical, it does suggest that, so far I've been talking about how if we knew positions and effect sizes of selected loci, we can predict ME or FST or whatever along the genome, but can we go the other way around? So increasingly, there are a lot of methods in genomic inference would give output something like this, which is basically effective migration rate or something, effective migration rate scaled by something along the genome, and then would say, this peak corresponds to a barrier locus, but that's at the level at which we are. So, but can we go from here, which is this, which tells us how the effective migration rate varies along the genome, and ask, sort of just not just say, okay, a peak is a selected locus, but actually use these kind of theoretical predictions to say that what is this sort of ME profile consistent with, make somewhat more detailed inferences. And of course, it's very likely that this is not going to yield, I mean, if I tell you this, I'm not going to be able to say, well, it means that there's one locus with effect size this, one locus with effect size this, but it does provide a sort of relatively economical way of saying, what is the family of architectures that this sort of ME is consistent with? And I think that's a sort of interesting direction to take. But as a caveat, I do want to say that, a lot of these inferences of how ME varies along the genome, how they actually are themselves based on assuming an underlying neutral model and sort of treating the effects of selection, encapsulating them via an effective parameter. And as we sort of saw, that is valid only when, this linked selection is a fast process. So how reliable are these estimates of ME given that they are sort of based on these kind of assumptions? So I think, as a sort of broader point, I think there's quite some scope here for going back and forth between theory and inference if we want to understand how these different loci sort of together shape patterns of diversity along the genome. Okay, thanks.